Now, today we will discuss about anti-cancer antibiotics. Now, before going into any details about anti-cancer antibiotic, we must know that what is this concept of antibiotics. Now, antibiotic, this term might suggest simply that they are against some biomass. Now, biomass means that can be any kind of cell or tissue, cell or tissues. But there is more to it. It's not just anti-biomass, because there are so many different things which can be termed as anti-biomass, but they do not fit into the definition of antibiotic. Now, let's consider a simple substance, phenol. Now, phenol is something which is widely used for almost a century as antiseptic. Now, antiseptics are also some chemical substances which will be killing different type of cells or tissues. Now, what is the then major difference between an antibiotic and antiseptic? The answer lies in two different points. The first point is origin. In most of the case, what we will see is that antiseptics are originated in laboratory. That means they are synthetically obtained. Whereas on the other hand, if we are thinking about antibiotics, they are obtained from some other type of microorganism. Now, these microorganisms can be bacteria, these can also be fungus. So, it can be bacteria or it can be different type of fungi. And the second important thing in case of the definition of antibiotic is that the amount required, or in other term we can say concentration required. Now, in both the cases, what we will see is that though they can kill cells, the amount or concentration of antiseptics which is required is a few thousand times more than antibiotics. And also another important thing which we should remember about the difference about antiseptic and antibiotic is their mode of action. Antiseptics basically they work topically or maybe inside a cavity. That means most of the uses are non-systemic external use, whereas in case of antibiotic these are systemic type of use, and to some extent there can be topical administration of antibiotics as well. Now, preferably antibiotics are targeted for microorganism, preferably bacteria. Now, there lies a question that why then this term anti-cancer antibiotic is being used here. Now, anti-cancer antibiotics or any kind of drugs which we know about, there is a concept of selectivity. Now, the term selectivity means how much it will go, say, inside a bacterial cell, and how much it will go inside human, or in this case we can use another term, host cell. So, this is called selectivity. If we are talking about an ideal antibiotic, they should be more selective towards bacterial cell and less amount of selective towards human cell. However, there are certain drugs which are less selective to bacterial cell and more selective towards human cell, or we can say both have equal type of selectivity. Such molecules can actually be used for the purpose of prevention or killing different type of bacteria, not only bacteria but human cell, especially in this case cancerous cell as well. Now let's talk about the different points and facts of the anti-cancer antibiotics. See, anti-cancer antibiotic have many different type of mechanism of action. These mechanism of actions can be related to the DNA strands. Either they will be directly attacking the DNA strands, and by this process they will be creating a spurious DNA strand, or maybe by creating some kind of highly reactive substances which will also be reacting with the DNA strands. In short, we can say in both of the cases they will be interfering with the S phase of a cell cycle. Next up, what we have is that let's just quickly discuss some major type of important facts related to anti-cancer antibiotics. So, as I told that antibiotics are either naturally or semi-synthetically derived. That means there might be some kind of chemical intervention in the process of the synthesis of these kind of antibiotics, but majority of these antibiotics are synthesized inside a microbial cell. In the next slide, where I have given some of the major example of anti-cancer antibiotic, we have discussed about these type of microorganisms from which these antibiotics are derived from. Obviously, semi-synthetical type of substances are also there, but in a majority of the cases or the approved drug as anti-neoplastic antibiotics, we can see that they are obtained from biological source. Remember one thing, that if certain substances can be obtained biologically, generally there are preferably no synthetic route, because if you see the structure of these anti-neoplastic antibiotics, they are so large in nature that chemical synthesis of such substances are nearly impossible, and even if it is possible, these will not be financially viable, thus making this type of drugs extremely costly, than which is already which is a cost which is viable for, say, a biologically grown or biologically derived substance. Now let's talk about their action, that these class of drugs generally affect DNA synthesis and replication by inserting into the DNA or by donating electrons that results in the production of highly reactive oxygen compounds. This concept of reactive oxygen compounds are very interesting. They are also known as they are also known as reactive oxygen species, reactive oxygen compounds or reactive oxygen species. They are very important at different conditions. So, they play very vital role in other parts of our cellular mechanism, and at times they can create harm to normal human cell, and that is why you can understand that these superoxide or reactive superoxide is a major reason why anti-cancer antibiotics has its major amount of side effects. You will learn about these side effects later in your pharmacology class. Next, what we have is that they are mostly exclusively given in an intravenous infusion process. Oral type of administration is not generally preferable, because all the gastric substances, they will be degrading the highly sophisticated products, and thus the uptake, the final bioavailability of this drug will be extremely low, and this is why generally they are given in this format of intravenous infusion. It is also used for the treatment of lymphoma and leukemia, and generally for other type of drugs such as nephroblastoma, known as the Wilms tumor, sarcoma, and cancer of the testicle, breast, thyroid, lung and stomach. That means these type of drugs have a high spectrum of use, and these can be used for such a long list of cancer which is written here. Now let's see some of the structure. Remember that these structures are little complicated, a little larger in structure. Dactinomycin and bleomycin, these two type of drugs which I have shown you here, they both occurs from Streptococcus species. Actually all of these drugs generally they are found from Streptococcus, even in this case of in this case also. So particularly streptomyces, and these type of drugs are generally called streptomycin for that reason. Streptococcus or strepto, let me write as streptomycin. Let me clear this thing. Yes, yeah. So as I was discussing, that they are obtained from Streptomyces sp. So the same goes for the daunorubicin as well. You can say doxorubicin, daunorubicin, both of them they belong to something called the tetracyclines. Now tetracyclines are also obtained from Streptomyces sp, Streptomyces species, and on the other hand there are other drugs which have larger structure. It will be important for you to know the different structures of these drugs, as they will carry importance in your final examination. Especially if you look that daunorubicin and doxorubicin, there is a very interesting fact which I am going to tell you, is that daunorubicin is found in a lot of different type of bacteria or wild type of strains of Streptococcus, but there is only one species of Streptomyces which is known as Streptomyces paucitus, and this is basically used for the purpose of anti-cancer. Both of them are used for the purpose of anti-cancer, and in this case you will see the major differences between these two drugs are basically one, lack of hydroxyl group in this position and the presence of hydroxyl group in this position. So if we look into the differences, so there will be such things which are present. Next type of drugs which we are going to discuss here is known as the plant products. Now plant products, obviously there are many different type of plant products which are present, and the plant products which we are talking here is not just one type of drug, and there are quite a few number of drugs which can be combined together and discussed as plant product. Here we will be exclusively discussing for the first type of plant product here, which is known as the vinca alkaloid. Now vinca alkaloid is a very common type of anti-cancer drug, and it is widely used in many different type of cancer. In this case we will see that these type of plant products, they will have their own mechanism of action individually. So first case, this vinca alkaloid has a very peculiar type of a role. Before going into what exactly the role is, we need to remember that what was the type of cell division process in case of mitosis. Now in case of mitosis in an animal cell division, we knew that, say if this is the cell, and in this cell suppose there has been two different chromatids which are present in every single case. These are called the sister chromatids, and these sister chromatids, now after the process initial phase, these sister chromatids has to detach from one another so that they can have or they can form two distinct cells. Now first, say there is one particular type of organelles which is present inside an animal cell, which is known as centromere. These centromere, what they will do, there is something called the centrioles. they will create something called the astral ray, which is basically made up of microtubules, and these microtubules, just a moment, these microtubules will basically pull these type of chromatids in two different directions so that it can further proceed into the cell division process like this. For example, after this process there will be like this 2 3. So one part of these chromatids are now differentiated from one another. Now they can proceed with the fissuring process, and then two new cells can occur. So this is a very basic thing that happens in case of mitosis in case of animal. However, it is obviously different in case of plant. So these type of alkaloids which are present in plant, they are known as the tubulin inhibitors. Now you can see that they will be inhibiting the formation of these protein called tubulin, which will form the microtubules, and if the microtubules are not formed, then obviously the result will be in such a way that these cells will not be able to divide, thus creating or halting the process in between. So these type of drugs, what we see is that it is a very commonly type of drugs which are obtained from a very common plant which is known as Catharanthus roseus, also known as the pink periwinkle plant. Pink periwinkle plant, it is commonly found in our regular home as well. In Bengali it is called Noyontara, and there are, I think, so many other different names in regional languages. So there are many different variant of this plant, but generally what we see is that the pink variant is where we get these drugs from. So it is naturally extracted and not much of chemical treatment is required apart from the separation process. We see there are 4 major important type of drugs which are important here, and these drugs are known as vinblastine, vinorelbine, vincristine, and vindesine or vindesine. So these type of, at least there are so many other type of alkaloids which can be obtained from the periwinkle plant, but not all of them are equally important. These are the two structures of vincristine and vinblastine. As you can see that these are, as these are plant derivative, they have a very detailed structure which is present in both the cases. In case of vincristine and vinblastine, it will be very important for all of you to practice these structure, at least even if you do not want to mug up the structures, at least try to know the features of each of the drugs. But even if you want to know the feature of each of the drugs, you will need to practice at least 4 to 5 times so that you can have some understanding about this these type of drugs. So today's lecture will be up to this point, and on the next day we will be discussing about some other plant product, and apart from that we will also discuss about some different type of anti-cancer drugs. So we guess that by next lecture we will finish this topic.